Hello, everyone. This is Jeff Bockman, EVP, Head of Oncology at Lumanity BioConsulting. I'm honored to be speaking today with Laurent Levy, Head of Nanobiotix. Nanobiotix is a fascinating company with a very unique type of approach for treating cancer, and I look forward to the discussion with Laurent. We've been working together for many, many years, and it's, I'm sure, going to be a very stimulating discussion. So, Laurent, you want to give a brief intro about the company and the platform and the program?
Sure. First of all, welcome everyone, and thank you for giving us the opportunity to talk about immuno-oncology today and more things. So just about Nanobiotix, we have the aim to treat and help millions of patients with our product, and this using nanophysics. So we are a late-stage clinical company that wants to help patients from local cancer treatment to metastatic disease. We've been working for quite some time and have been treating more than 350 patients across different indications. We also completed 7 clinical trials, including the proof of concept showing that the mode of action of this product is working, and that in the randomized phase 3 clinical trial. We today have another global phase 3 ongoing in locally advanced head and neck cancer patients. So how does this work? We are using nanoparticles that we inject directly into the tumor in order to maximize the concentration and the efficacy in the tumor and minimizing toxicity and systemic exposure. This product is made to be combined with radiation, activated by radiation. When you inject the product in the tumor, it will go into the cell, then the patient will receive a normal dose of radiation therapy, and this product will absorb the energy and will deliver within the cell a high quantity of damage, killing physically the cell. So the primary mode of action of this product is literally physically destroying the cancer cell, but there is some subsequent biological impact. And this, by killing physically the cell, you trigger an immune response, you prime an immune response, and not from any specific biological pathway. You have many potential biological pathways activated with that because we don't target one specific biology. We just kill the cell and trigger immunogenic cell death, activating the STING pathway and many other things. What is interesting is not only we've been showing many preclinical data showing and demonstrating that, but we start getting some very interesting results into our phase 1/2 clinical trial, where we've been treating both naive patient to PD-1 and refractory patient to PD-1. And let me just take an example. We've been able to treat a patient that have priorly received PD-1, that got a response under PD-1, that progressed again and entered the trial. And after the treatment, we've been able to go back to a response for some duration. So what it is showing is not only we can maybe transform the refractory patient into a responder, but the fact that we've been able to come back from a refractory patient triggering a new immune response may be very interesting for a wide number of patients receiving checkpoint today.
That's great, and so you brought up a lot of points that I want to follow up on, and I'm sure the attendees would be very interested to hear about. So let's talk about what the data are, the preclinical experimental data that kind of support this idea of the ability to essentially, in what you just described in the clinical data to date, enables the program to convert what are essentially non-responders to a checkpoint inhibitor, to an anti-PD-1, PD-L1 agent, into responders.
So we've been internally, but also through collaboration, including a large preclinical collaboration with MD Anderson Cancer Center, we've been working from the basics, which is what the product itself is doing, and then working in combination with different type of checkpoint inhibitors to see how can we impact different type of combination. So let's go back to product activated by radiation. We've been running this critical and simple experiment where mice have 2 tumors, one on left, one on right, and you have an impact. You try to irradiate the left and see what's happening on the right tumor, which is not irradiated. So when we've done that in some of the experiment with radiation alone, you've seen the irradiated tumor shrinking, but the right one was growing. When we did give the same dose of radiation plus the injection of nanoparticle, not only the primary tumor did shrink, but the other one shrink too. And what we see here is in an animal without any checkpoint inhibitor, we are able to trigger an immune response coming from an irradiated tumor to a distant tumor. And that's technically what we would like to achieve in human. Of course, this is an animal model, so it has a lot of bias, but that was the primary experiment. And interestingly, when we look at what's happening in a non-irradiated tumor, we've seen massive infiltration of CD8. And when we tried to deplete the CD8, then there was no more efficacy. So it's purely mediated by the CD8 action. And that was the first set of experiment we've been doing. Then we moved to more complex model in combination with checkpoint inhibitors, and especially we've been taking models that are resistant to checkpoint inhibitor. So we've been able to crack different type of model using PD-1, anti-CTLA-4, LAG-3, TIGIT, different type of checkpoint inhibitor, and always have been able to improve and to change what the checkpoint can do, including something which is critical for us and for patient, obviously. It is the possibility not only to trigger an immune response, but to have a long-term immune response that can ensure that we will get some memory T cells and therefore long-term for recurrence of the disease.
Great. So even in models, it's been challenging for many companies to show that abscopal effect, which is great. I think you even have some ability to infer that that might be occurring in some of the clinical studies that you have ongoing. We'll circle back to that in a bit. I'm glad you just mentioned the issue of the long-term memory in terms of memory T cells, et cetera. Maybe you can go a little bit more into that. Have you actually done re-challenge experiments where you are demonstrating that those mice can be protected against presenting them with the same or sometimes even with a different tumor type?
Sure, and this is obviously essential. So yes, we have been taking and keeping the animal that survive with complete response with a combination of radiation plus nano plus some checkpoint inhibitors. I've waited for a while, then I've re-challenged the animal with new fresh cancer cell, not seeing any growth of tumor in those animals.
Great. So, it strikes me, that it's really interesting, not only because of what you're showing and not only the kind of let's call it the holy grail of being able to turn non-responders into responders. Obviously, the preclinical models are supporting the story there. And, we'll talk a little bit about some of the clinical data. But I think it also should be emphasized that with this physical approach, you are essentially kind of tumor agnostic without having to, be complicated about it because, as you've said, this is physics that is at work. But it's very much orthogonal to many of the other approaches that one is considering. And one might believe that orthogonal approaches are especially useful in a situation where many biologic pathways of resistance might be enacted against a checkpoint inhibitor, whether that's innate resistance or acquired resistance. You want to talk a bit to that?
Sure. I think it's orthogonal but not incompatible to start with. And you're right when you say most of the time when we try to target a biological pathway, whatever is this pathway with the treatment we are doing, including some of the checkpoint, biology always finds its way to go over one or two or three pathways. So an option is, and that's what we're doing, is get rid of the pathways, at least at the primary mode of action perspective. If you kill cells from many ways, triggering many types of subsequent biological pathways, then you trigger, let's say, not only one immune response, specifically activated or mobilized through one or two pathways, but through many pathways. And that's where you can see that when you start with a patient that had an innate immune response and got a benefit from checkpoint, then stop responding, if we can take this patient and make him respond again, is because we have triggered an immune response that was not here before. That was not naive immune response. So here is what we show is there is a possibility through physics to kill the cell in so many different ways that we could trigger not one immune response, if I can say, but many immune responses. And I think that's where the physics could play a major role in oncology.
Yeah, so this is very much like kind of the optimal situation for in situ vaccination, for that immunogenic cell death, the release of all the antigens, including neoantigens, as well as all the other PAMPs, DAMPs, inflammatory signals, everything designed to kind of bring in, recruit, activate various arms of the immune system. Of course, still dependent on kind of de-repressing that tumor microenvironment with a checkpoint, if not with other agents as well. And you did mention a range of other agents that you've been exploring, at least preclinically, LAG, TIGIT, et cetera.
Yes. And what we have been showing too using the nanostring technology is when you look at the non-irradiated tumor in some of the experiments we've been running, and you look at all the adaptive immune response pathways, it's not only one or two that are lighting up, it's the entire range of adaptive immune response. So it shows that we are not working with only one path, but many passes.
Great. So I'd like to touch a little bit more on the clinical data and then talk a little bit about kind of the business model and the business of nanobiotics in a world where there is so much IO noise, ongoing. So, but before that, let's just, touch a little bit more on kind of that clinical data that you're generating and some of the other ongoing studies, the additional readouts of the 1100 study, and some of the other studies that you're planning.
So first, I'll come back to what I say in the intro, which is we try to help patients on a broad spectrum, and starting with patients that have locally advanced tumor, where in many cases, they don't need a checkpoint inhibitor, but they need something better from a local perspective. And here, the combination of radiation plus our product could play a seminal role to help them. That's what we have shown in the soft tissue sarcoma phase 3 trial. That's what we are intending to show in our new locally advanced head and neck cancer patient phase 3 global trial and many other running trial. Now, if we go on the other side of the spectrum with metastatic patient, where checkpoint inhibitors play a key role in this, we want also help patients. And that's where we start developing different clinical trial alone or within our collaboration with the MD Anderson to try to see how big could be the impact of this product for those patients, both from a local perspective, but more importantly, from a systemic perspective. And we've been running a clinical trial that take either naive or resistant patient to PD-1 that have either a locally recurrence of head and neck cancer, a lung met, or liver met coming from any other primary tumor. What is interesting here is that when you look at literature, depending on the series, you can see that the patient usually could have a naive patient from 15 to 40%, depending on which indication you're looking at. But let's say that within the head and neck metastatic patient setting, then you have around 15% responder from the naive patient. So what we try to do here is not only to get a better, deeper response for those 15% of patients, but more importantly, the 85 remaining % of patients, we try to bring them some solution. And if we can do that, then we'll play a critical role. We've been treating in our trial a number of those patients. The only caveat is that it's still a small number, so we need to expand, and that's what we are doing now in this clinical trial. But in the number of patients we have treated, we've seen something very interesting. Is that like in a refractory patient, we got an overall response rate, so systemic control of the disease of 36%, which is more than what we could assume in a naive setting population. So of course, this has to be confirmed in a broader population, but imagine that this is confirmed. So it will mean that adding in non-responder, the product plus some fraction of radiation could really change the face for many patients receiving IO.
Yeah, and this is, again, to use that phrase, holy grail again, the idea of kind of broadening and deepening the response to checkpoint inhibitors, and especially in those who are progressors, whether they are kind of non-responders or ultimately respond and progress. So that's really very tantalizing clinical data so far. So let's switch a little bit now to let's call it the kind of the BD side of nanobiotics. I mean, clearly we are in a world now where there suddenly is a lot more interest in radiopharmaceuticals, number of deals and startups, et cetera, kind of reinvigorating that space. And those are probably in part also abetted by the reinvigoration of antibody drug conjugates. But I think it's important to distinguish, how Nanobiotix is different from many of those other companies. And one thing I would note is that, because I don't think you actually mentioned it, is that this is a one-time injection
Correct.
of the product of NBTXR3. So, but maybe you could talk a little about kind of how you see yourself positioned in this space of kind of radiopharmaceuticals, and also kind of what the reaction and how this story is gaining attraction or traction within the biopharma industry.
So first, I think we are not a radiopharmaceutical because we have not a radioactive product, but we're still possibly we could be combined with radiopharmaceutical because everything we need is a source of radiation to activate our particle. The main difference here is, let's say, for example, you took head and neck cancer patient or lung cancer patient, stage 3, they're mainly not going to receive radiopharmaceutical, because there is already a standard of care that is giving radiation, chemo when eligible, plus some checkpoint inhibitors and so on. So the radiopharmaceutical will have a lot of problem to enter this field because they will have to compete with a strong standard of care. With our product, we don't compete with the standard of care, we just insert ourselves in a standard of care. So let's say that in general, every single patient receiving radiation at some point could benefit from our product, either to get a better efficacy or to get a lower toxicity. So this excludes the part of patients that do not get radiation today, where we could improve the use of radiation in that. But that's how we see things. And a lot of competition in the IO or metastatic setting with patient, and much less, if not very no competition in the early stage of cancer treatment. Now, what is very interesting is that we have seen first a move from the medical community. And this happened years ago when we did publish our first positive phase 3, establishing in soft tissue sarcoma, which is a very hard to crack disease, the mode of action of this product, showing superiority by adding nano to radiation. 2, for the past 12 months, we have seen a growing interest from industry looking for a new solution that are not just a multiple combination of different checkpoint inhibitors, where we know that combining biology to biology bring more uncertainties, more risk, and diminish the number of patients potentially that could get value out of it. So coming back to basics of treatment is, from what we see, an interest that is growing into the pharmaceutical industry these days.
Great. And actually, we'll look forward to having a discussion at Cancer Progress about this idea of the many combinations, because there are published data. And in fact, I'll have a discussion with Emmett Schmidt about the actual evidence of combination value and synergy, because there's some argument that, many of these studies, even if they've shown a signal, may not really be a signal that's meaningful of true biologic synergy, which is a different situation for you, given the mode of action of R3, which is great. It does beg the question, though. I'm curious because, having been around, as you can tell by the gray hair, for a long time, there's been a lot of ups and downs with this idea of kind of radio enhancers. Do you embrace that term? Do you reject it? Do people use it as a positive or pejorative when you're interacting with prospective partners or others, investors even?
I think, Danny, in this, we should clearly differentiate radiosensitizer versus radio enhancer. There have been many attempts and some successful. If I take cisplatin as an example, that a super radiosensitizer, when you combine with radiation, you get synergies. But it's a biological compound or a chemical compound, so you target something with it. So I think the radio enhancer is different from a radiosensitizer. A radio enhancer will enhance the dose of energy that you will deposit into the cell. So will not be linked again to any biological pathways. And the fact that you do that directly into the tumor through an intratumor injection is the best pathway you can imagine. When you get cisplatin, into a patient that has systemic exposure with the toxicity we know. And if you have a local cancer to treat, then you get a systemic exposure and systemic toxicity for local benefit. Obviously, cisplatin works also for MET, but that's another story. A perfect radio enhancer should go directly into the tumor with all the product in the tumor and not going outside the body. Then you maximize the efficacy and you minimize potential side effect. So there is two different pathways, and so far we're the first and the most advanced radio enhancer in this world.
Yeah, which I think is a great value, but can also be sometimes a little challenging to be the unicorn and kind of educate everyone about, why you're unique and special.
Always, but that's the price to pay if you want to disrupt something.
Yes, and I think disruptive is a great way to kind of move towards closing out the discussion. So I think we maybe have a minute left or so. So what would you like to leave the audience in closing to think about in terms of Nanobiotix and the lead program, NBTXR3?
So we're moving forward in many indication, but our priority is this global phase 3 running in locally advanced head and neck cancer patient and the expansion of our product into head and neck refractory to PD-1 patient. And now we've been discussing with FDA beginning of this year in order to design what our pivotal trial could be within this IO combination field, and we will update everyone on that later for sure.
Great. Well, we'll all be looking forward to that. Certainly I will. All right. Thank you so much, Laurent. It's been great talking with you. Thanks to everyone who's attended this virtual session, and hopefully we'll be seeing each other soon in the real world. Thanks so much.
Thank you, Jeff.
Thanks.